Oop, there we go. All right, welcome back, everybody, now that we're recording. So we're going to start with our first presentation, which is on the mutual influence of human and artificial life and experimental investigation. And I don't know if I need to enable anybody's audio or if they can do it. No, okay, good.
It should all be working. Oh, we cannot hear you, Eric. Seems like. No, nothing's getting through. Seems like there's some inputs. Maybe the setting is not right for the inputs. Sometimes Zoom gets confused. Still seems nothing is getting through. Can try the good old unplug and plug.
So sometimes there have been problems where we can hear people, just not if the slides are going. So we might want to see if it's a general problem or if it's only a problem when the slides are being shown.
Bit strange, yeah. Yeah, you can try, Eric, to try to stop screen sharing, see if the sound comes through then. Because there has been a weird bug like that, as Lisa just said. But it seems like...
Nope.
Yeah.
Maybe leave and rejoin.
Yeah, exactly.
Maybe that's what he's doing.
That was the next one on the list. If it doesn't work, I guess then we can just swap and have the next one, or like start with the recorded talk, and then see if Eric can.
Now, do you hear me?
Yes, we hear you.
Okay.
Awesome.
Super nice. Okay, just a moment. I share the screen again. Do you see the screen?
We do, and we still hear you. That's great.
Okay, perfect. So I can start. Sorry for the inconvenience. Okay, so I'm Eric Medved from University of Trieste, and this work is the result of the joint effort of Stefano, a master student, Georgia, and Federico, two PhD students. So most of you probably knows this video. It's part of a series of videos in which basically there are a few guys that treat badly some robots. Actually, this is fiction. Okay, no real robot has been beaten for doing this video, but we all have these kind of feelings, these kind of questions arising inside ourselves about the fact that there's what is happening here. Is the robot suffering? What are the feelings of these robots? Maybe we also think about future robots, maybe descendants of these robots seeing this video. Will those descendant robots be somehow affected by this behavior? So more broadly, there will be an impact of this kind of actions on the species of robot that is somehow developing in being, let's say, inserted right now. Fortunately, this is just fiction, so no real robot has been beaten, and we actually don't need to answer this question. But there are some kind of agents, with artificial agents, with which we interact constantly and actually in our real life. Some of these agents, maybe, let's say, very complex systems, are indeed also plastic in the sense that they can change in response to environmental conditions. And we, as humans, are part of the environment of those agents. And in such a way, we can potentially impact on the change these agents can go through. Some of these agents has been also designed right with the purpose of being able to change in response to interaction with humans. Here we see a significant example. Some years ago, Microsoft designed and released a chatbot for Twitter. Basically, this chatbot was designed to be able to interact by means of tweet and replies written by means, of course, of natural language with humans, and also to learn from those interactions. What happened is that at the beginning, the, let's say, personality of the agent was quite nice and sweet. Okay, so as you can see in the two tweets, two examples in the top row in the slide, this agent was quite nice. But just after a few hours, right, as a consequence of the interactions with humans, the agent turned out to be, let's say, a very bad personality, a very bad guy, okay? A sort of Nazi, anti-feminist, extremist. So this experiment is an example that artificial interaction of humans with artificial agent may matter. So a couple of very big questions arise. Do we humans behave well with artificial life forms? Is artificial life form somehow adaptable, somehow influenceable by humans' actions? And this means that it has to be somehow adaptable. It should be able to change in response to interaction with humans. And provided that it can be influenced, are we humans aware of our potential influence of artificial life? Of course, these are huge questions that are very hard to be answered for countless reasons. What we try to do in this, let's say, humble study is to give a sketch of answer following somehow a sort of scientific method. Okay, so what we did is basically to follow this recipe. First, build a system and then study it. In particular, first build an artificial life system that is suitable for trying to give an answer to these big questions, or a sort of sketch of answer. And in particular, this means that this artificial life system should be influenceable, potentially influenceable by humans, allowing for interactions, and adaptable in such a way that it can change. Okay, and of course we needed to define what are interactions, and then design an experiment allowing to this interaction to happen, measuring and observing them, collecting them, and analyzing them. And finally, we needed to define what is the influence on both sides of humans on artificial life and of artificial life on humans. And of course, according to this definition, to try to draw some conclusions. So the largest effort in this study was devoted to the design and development of this artificial life system because of its peculiar requirements. In particular, we wanted a system, an artificial world that looks alive. Not necessarily like real life, okay, not necessarily like, let's say, biological life, but something that seems alive. And we also need to take in consideration the fact that most of humans today are somehow familiar with very advanced form of simulation of creatures, basically video games. Okay, so we are somehow in the middle of familiarity with great complex, with greatly realistic form of life, and the need to have something that actually looks alive. Second requirement is that the artificial life system should allow interactions in terms of information and so causality moving between the artificial life world and the real world, and the opposite. We needed the two kinds of interaction. Note that the second one is usually more peculiar, so that the way that humans should somehow impact on the artificial life system is a rarer, let's say, requirement. Finally, we needed a system that is adaptable, that has the potential to change over time. And finally, we wanted everything to be simple enough to be understandable, so possibly explainable, because we did some experiments and we wanted to exclude, let's say, second order motivations. So what we decided to do is to build an artificial life world in which there are a few worm-like creatures hunting for food. We chose this form, this shape of artificial life because they somehow, let's say, resemble something that is alive, but is not something, let's say, too complex. In particular, we wanted to give this idea of a human observing a small world with microorganisms moving in a, maybe through the lens of a microscope, moving in their world, something that is close enough on which you can interact, but not exactly where you are inserted in as a human. So we wanted these worms to look alive, and for this reason, they should have been, let's say, capable of doing something, okay, not static, so basically of moving, but possibly of moving according to some let's say, criterion, possibly hunting for food. So for achieving this goal, we made them capable of sensing the environment through 4 senses: sight, smell, touch, and temperatures, to process the perception they acquire from the environment through an artificial neural network, and to actuate, to somehow apply the result of this processing. And this is done in the sense that they can move. Basically, they can go ahead with the head or turn the head. Since we want a system that is adaptable, first we need to have something that can change over the time. The first thing is that there is a life cycle in place, okay? And this is based on energy consumption. So basically, the worms consume energy just by living with a constant rate over time. And they are able to acquire energy by eating, so touching basically the food items. Whenever a worm energy goes to 0, basically the worm dies, and instantly we add a new worm in the artificial world. So in such a way that we have a constant number of artificial agents in the simulation. We also, to keep the balance of energy, let's say, constant, we also add some food when a worm dies. We use the life cycle to make the system actually adaptable in terms of an evolutionary time scale. So what we do is that worms can differ in terms of their morphology, so number of segments. You saw that they are composed of small circles, the number of, let's say, legs, flagella, the color, and also they differ in the brain. Okay, so the neural network that actually perform the processing of perception to give actuation. All of these traits are encoded in genotypes, and whenever a worm has to be generated, a new worm has to be generated, the genotype is inherited from some of the existing alive worms. By doing that, and by imposing a criterion according to which one worm is chosen to be the parent of the newborn worm, we basically inject some evolutionary pressure in the artificial world. What we decided to do is to base this pressure on age, on the assumption that the long-lasting, the more, let's say, the older the robot is, and very likely the better is its ability to be alive, to hunt for food and everything. A key requirement of our artificial world was that it should have allowed interactions. So we can categorize interaction from the real to the artificial world, and from the artificial world to the real world. Or from another point of view, this is the direction of the flow of information going from one to the other side. Concerning the first case, from real to artificial, so what humans can do on the artificial world, we have 2 kinds of interactions. One is explicit and one is implicit. Concerning the explicit one, we allow the human to click on the simulation, so basically with the pointer of the mouse on a graphical representation of the artificial world in real time. The human can click on the simulation, and depending on what he or she clicks, the outcome is different. He can add a piece of food clicking not on a worm, or killing a worm clicking on a worm. Also, we allowed for an implicit interaction. Whenever there is a human looking at the simulation going on, through a webcam, we acquire the presence and we change the temperature in the artificial world accordingly, in such a way that the worms have somehow the perception of the presence of the human looking at the simulation. On the other direction, basically the information flows from the artificial to the real world by means of the fact that humans can visualize the ongoing simulation. As you can see, there are two kinds of explicit actions. One basically is a good action, feeding the worms, and the other one is a bad action, killing them. We decided to have this very sharp division in order to facilitate further analysis of the experimental results. So we can basically very easily classify good humans and bad humans depending on the kind of action they perform. One thing that we needed to do, of course, is to be able to measure in the influence. Also, in this case, on the two directions, so influence on the artificial life from humans and on the other side. Concerning the former, what we did is to perform a few long-term experiments in which basically we simulated a few humans. The reason for which we simulated them is because of the duration of these experiments. We could not involve real humans for several hours, okay? So we simulated good or bad humans or no humans at all, doing accordingly good or bad actions. And we kept note of some, let's say, worm metrics, basically age, travel distance, size, and so on, in order to be able to see if something is happening. Concerning the influence of artificial life of humans, what we did was to perform a user study in which basically we collected the answer to two questionnaires, one given before and one after the interaction performed by real humans, real subjects with our artificial life world. So let's see the result. The key result is the one concerning influence of humans on artificial life. Unfortunately, we were not able to get a good result in the sense that our experiments, despite the fact that we designed them and the simulator in such a way that we should have been able to see the influence, we saw no influence. In this figure, you can see the values of three key indicators, three key metrics. So the average age, the average travel distance by worms, and the average size of the worm during the long-term simulation of the artificial life world with or without human actions, with two cases, good action and bad actions. As you can see, there are no clearly visible differences. So we cannot say if with this simple setting there is an influence or not. We also thought about possible causes of this, let's say, missed result. One is that the experiment was too short. One is that very likely the way we build the simulator is such that both good and bad actions of humans actually relieve the evolutionary pressures, making basically food hunting an ability that is not advantageous. So there is no more pressure and nothing can be observed.
And we have 2 minutes left, by the way.
Yes, I'm almost done. Concerning the user study, we involved 36 participants of different age, different familiarities with artificial, let's say, agents, video games, and real biological life forms like pets, okay? And here I'm going to present just a few results, the key one. The first thing we try to investigate is whether humans believe in artificial life or not. So as you can see here, we asked them, do you think artificial life exists? And we also try to correlate somehow this answer to the question, to the answer of the question, how would you rate your perceived involvement? What we see here that most of our subjects believe artificial life exists and were indeed involved while interacting with our artificial world. A second big question is about the actual behaviors that humans had, so good or bad actions. The nice thing is that no one planned in advance to be bad. So no one said, I will kill a lot of artificial life forms. Actually, they behaved accordingly. So just the one without an opinion performed a few more bad actions, okay? And finally, we also investigated about the let's say, what humans think about the possibility of artificial creatures suffering from bad actions. Basically, as you can see here, most of the humans, the good humans, let's say the good humans thought artificial creatures can suffer and behave accordingly without make them suffering. The mostly interesting thing probably here is that the humans without an opinion about possibility of artificial life to suffer are the ones that behave, let's say, worse. We also investigated about the possible causes, in particular, why did you kill an artificial life form? Why did you kill a worm? Most of the humans said by mistake, some for curiosity. Somehow, someone told us, because I saw that worm was not good enough, and so I decided to kill it. And this is somehow disturbing. Okay, so summarizing, what's the take-home message? Our society is becoming more and more complex, with more and more interaction between real biological agents, in particular humans, and artificial agents. And of course, there is an emergent interplay, but studying it systematically is very hard. This is just an attempt. What we did is just an attempt to apply, let's say, a scientific process to capture this emergent interplay. And somehow we failed, but I think we found some interesting results. One of them is that, according to our subject and to our user study, humans are potentially sensible to artificial life. And this is somehow promising, in my opinion. Okay, thank you very much. I'm done. If you have any question, I'm available.
Great, thank you so much. So if anybody has questions, we don't have time to take them live right now, but you can either direct message them to Eric on Slack or here, and we're going to move on to our next talk. Okay. So as Richard mentioned, our next talk is recorded, so we're just going to go ahead and play the video, but the talk is called The Evolution of Fractal Protein Modules in Multicellular Development.
Hi everyone. Today I'll be talking about our research, the evolution of fractal protein modules in multicellular development. A fundamental process in the controlled formation and development of plant and animal phenotypes is regional specification, also known as pattern formation. For example, in human development, the basic body plan of the embryo becomes established during a very short period from 1 to 4 weeks after fertilization. For a multicellular assembly, the problem is one of self-organization. Cells must differentiate in the correct spatial locations with no explicit knowledge of their position within the final assembly. The diagram shows how a single cell becomes an undifferentiated multicellular organism before each cell then switches to the appropriate cell type through some mechanism in order to achieve a targeted flag morphology. This classic morphology is known as Wolpert's French flag, which was introduced in 1969 as a simplified toy problem. Whilst there are various theoretical models which aim to describe how patterning occurs in biological development, some research activities falling under the name of artificial development attempt to gain insight to the problem using simulated evolution and abstracted in silico models of development. On the right, you can see a reference to a paper in which the authors integrated an artificial gene regulatory network with the cellular pods model of cell growth and sorting. Using simulated evolution, they obtained organisms which were able to autonomously establish morphogen gradients and use these to obtain the positional information needed to accurately reproduce the French flag. We follow a similar modeling approach using an artificial model of gene regulation within a cellular pods framework. However, our model of gene regulation is based on an artificial chemistry system called fractal proteins. Fractal proteins were first introduced by Peter Bentley in 2003 and are an example of an artificial chemistry. Like biological proteins, fundamental to the interaction of fractal proteins with any system is their shape. This shape is genetically specified by a triple XYZ, which points to a finite region of the Mandelbrot set. This subregion is referred to as a fractal protein. Now, as well as providing a compact representation of shape complexity, the geometry of the Mandelbrot set makes the fractal genetic space highly evolvable due to its continuity and cell similarity. So how does gene regulation with fractal proteins work? Well, starting from 1, the cellular protein interactions, At any point through development, there are a number of fractal proteins within the cell. At each development time step, these proteins interact to form new complexes through an artificial chemistry, an example of which is shown in Figure 4. We then follow the red arrow, we see that these protein products, which we call transcription factors, then go on to either promote or inhibit the expression of genes within the genome. This is based on a shape similarity between the fractal shape of the transcription factor and the fractal shape associated with a gene's promoter region. Expression of a gene results in the appropriate fractal protein being transcribed and its concentration therefore increasing within the cell. Depending on a type identifier, these then either trigger cellular development processes or take part in the next round of gene regulation through the production of the appropriate transcription factors. Every cell in our multicellular model is equipped with an instance of the fractal GRN. In the next slide, we explore this multicellular model. So our spatial model of cells is the cellular pods model. Introduced by Grainer and Glazier in 1992, it was used to simulate the spontaneous sorting of a mixture of two types of biological cells. The fundamental components of the basic CP model are a lattice representing a spatial environment and two functions, the cell identity function and the cell type function. Different surface energies are defined between cell types. This drives the sorting as the system moves towards an energetic minima. In our model of development, sublattices are introduced on which the varying concentrations of different fractal proteins are specified. In order to give the GRN control over the developing morphology, the integrated model connects the regulated concentration dynamics of the fractal proteins expressed by various genes with various terms in the Hamiltonian. As an example, we can link the target area of a cell to the concentration of a specific fractal protein. The cell will then scale in size as the concentration increases, hence growing. Now, in this project, we used a quality diversity evolutionary strategy called MapElite. Now, usually, evolutionary algorithms are focused on pure optimization, that is, finding a single high-performing solution. In quality diversity algorithms, the aim is to explore the solution space as much as find high-performing solutions. This has been shown in some problems to both improve the overall fitness and the variety of behaviors discovered. As well as bringing the benefits of QD algorithms to the search for good solutions, it also suggests a new formulation of the French flag problem and sets one of the themes of the research. Supposing that there are 4 possible cell types and hence colors, we can see that if we characterize this 4-color phenotype space, we obtain a set of flag-like morphologies, where each niche is therefore characterized by the number of stripes and the color composition of the organism. Importantly, it also creates a related set of problems with inherent regularities, 1 or 2 stripes, some flags both having blue patterning, some having green, etc. One question then explored in this research is, can evolution learn regularities in this new problem space and hence preserve and reuse parts of its solutions? We will revisit this question, but first let's see the results of evolution and see if we obtain any fit-like flag phenotypes. Evolution was run for 500 generations, and runs were seeded with a population of 10,000 genomes, each of which were initially initialized with 35 genes with a certain configuration. Now, organisms were allowed to develop for 800 time steps, starting from a single cell in the middle of the grid. Now, the first question of whether fractal proteins can coordinate temporal and spatial gene expression to reach a targeted morphology, the answer is yes. The results demonstrate clear examples of regional specification and spatial patterning. In particular, we found MapElite to be beneficial no penalization terms were added to the fitness function in order to encourage organisms early on in evolution to use all colors and hence avoid local minima. We also ran some other tests on the evolved organisms, aiming to assess the stability of the obtained morphologies and the robustness of the development process. To test for stability, we allowed evolution, the development time for run for twice that used in evolution, and then assess the percentage fitness change from the original development time. And for robustness, we randomly perturbed the internal concentration levels of a subset of cells at random points through development. Each of these perturbations was applied as a random increase or decrease in protein concentrations up to 10% of the original. And in each trial, we targeted 30% of cells. The results were mixed for both of these, although we did find it interesting that some did achieve both stability and robustness without it being an explicit focus in the evolution research. So going back to the general flat problem, we saw how our extended problem space now has regularities. In this paper on the left, Garibay described a new genetic representation which attempted to exploit regularities in checkerboard construction in order to scale performance better with complexity. His idea was to genetically encode something more akin to a construction procedure using certain functional primitives, draw and skip in this case, rather than a direct encoding of the checkerboard. Now, this procedure is really like a development process. Building this solution requires the use of certain subroutines used and reused at various points to produce the final solution. This idea is shown in the box on the right. This notion of decomposing a development process into a set of distinct developmental primitives or subroutines is connected to the concept of modularity in development. Key features of modularity is the reuse of subroutines to build a solution non-continuously, and the preservation and inheritance of these modules by related solutions, those that share some kind of regularity. We can explore this further in the context of our development system. In our model, development processes are coordinated through the expression of genes in specific temporal and spatial patterns. In our model, changes to a cell, i.e. development, are enabled by the activation of specific gene combinations. It is these functional patterns of activation which are considered the functional primitives or developmental subroutines that form our modules. The sequencing of these patterns, i.e. the order of A, B, or C, is precisely regulated by the evolved GRN. The precondition for expression of a particular pattern is the creation of an appropriate transcription factor. The transcription factor sequence, TF, therefore encodes the history of activation patterns used in development. To find evidence of module reuse, we therefore collected the full set of transcription factors produced through development for each FAG species and looked for common products. Figure 2 on the left shows a full set of 199 unique transcription factors observed across all organisms throughout their development. The color bar indicates the first time at which the transcription factor was detected within development. Figure 3 on the right gives a closer view of the common transcription factors, that is, the unique transcription factors which were identified in more than one organism throughout development. The existence of common transcription factors was the first indication that evolution may have reused parts of one solution within another. However, we needed to check the underlying gene activation patterns they were triggering. From the common transcription factors, of which there were 36, we examined the expression patterns of the genes making changes to the cell morphology. In the figure, we could see an example of a developmental module, We have the same pattern of activation triggered by the production of the same transcription factor in two species with regularities in their phenotype. In this case, they both have green and white cells. Now, in agreement with these final phenotypes, the gene activation pattern is associated with the proliferation of green cells in both organisms. We found that 17 out of 36 of the common transcription factors shared these characteristics, producing a common pattern of activation across the three behavioral genes in related organisms, but at different times. This was then strong evidence to us that evolution had reused part of its networks in constructing related phenotypes, despite the selection pressure of mapper leads to find novelty. So the conclusions, a combination of fractal proteins, cellular pods, and mapper leads has enabled us to explore the space of developmental solutions, successfully obtaining solutions which can develop flag-like patterns. In addition, some evolved organisms demonstrated desirable properties, such as robustness to environmental perturbations and the ability to maintain stable phenotypes. Crucially, evolution has discovered and reused patterns of gene activation, both within an organism's development, module reuse, and across different organisms, module inheritance, despite the selective pressure of mapper leads to find novel phenotypes. Our system therefore demonstrates the emergence of biologically plausible phenomena, such as the learning of regularities in solutions and the automatic creation and reuse of modules. We anticipate that these methods will enable further insights into artificial development processes in the future. Thank you for listening. Any questions, please ask on our Slack channel. It's been great presenting. Thank you.
Okay, great. And I think looking at the participant list, we do have Peter Bentley here. So if there are any questions, I think we do have time to take one or two live.
Yeah, I was going to say, Harry passes on his apologies for doing that as a recording. It was a bit of an emergency because he has no power at all at home right now. Because of the heat wave of the UK, the power got knocked out. So I'll do my best. If you've got some easy questions, I'll do my best to answer some. But otherwise, do ask Harry in Slack.
Yeah, that's definitely an unfortunate situation. I hope his power comes back on soon.
Yeah.
I was just going to ask if you happen to know what the behavior, it might have been in the talk, but I saw you used the mapper leads, the quality diversity algorithm. I was wondering if you knew what the behavior characterization was, or like I said, that might have been in the talk.
What specifically for mapper leads? That's a good question, and I'm not sure I know the answer to that. As far as I remember, he was just looking for diversity with respect to the colors in those flags. So yeah. So I think specifically he was looking for different types of flag and using mapper leads to explore that space. I couldn't tell you what the exact descriptors he used were there, unfortunately.
Okay, that's no problem. Thank you so much. Okay, it doesn't look like we have any other questions. Maybe some will get posted to Slack. All right, well, thank you. Let's go ahead and move on to our next talk, which is paths in a network of polydispersed spherical droplets.
Do you see my screen?
Yep, and we hear you, so you are all set.
Okay, thank you very much for your kind introduction. This talk is part of a 4-year project financed by the European Union. It's a collaboration, especially between the University of Trento, Cardiff University, the Zurich University of Applied Sciences, And we have also some external partners from Japan. The first two groups are mainly responsible for performing experiments, and our group is doing the theory and some simulations. So our question, which we regard now, is we want to see which paths are there in a network of polydispersed spherical particles, how many paths are there, how long they are. We deal with these questions for one specific reason. We have in mind to fill some droplets or vesicles with various chemicals, bring them in contact with each other in some network, and then perform a gradual chemical reaction scheme by allowing the chemicals to move through pores from one droplet to an adjacent another droplet. Here I present you results, especially for simulations. So I first of all explain about our simplified model for this agglomeration process. First of all, some spherical particles are randomly initialized in a container, which is a cylinder. They have some given radii, which are chosen randomly from some range. They are filled with heavy oils that they sink in water. They are within a cylindrical container with a proposed radius value, a proposed height value, and water value of the laboratory was also reused in the simulation. So we use the viscosity value at this temperature of 25 degrees. And at the beginning, of course, the particles are initialized in the way that they do not overlap and they have an initial velocity of 0. So it looks like at the beginning of the simulation. And then we have to perform the simulation itself. The overall simulation is divided in small time steps of 10 to the power of -5 seconds. And in each time step, each of the particle is subjected to some forces. So first of all, of course, the gravity force reduced buoyancy, then the Stokes friction force, and additionally we added the added mass effect, which is a little trick in order to also include some basic hydrodynamics. Furthermore, we implemented also small random velocity changes in order to meet the conditions in which the experiments are made, and also some almost elastic impact. So if some particles touch each other, then there are some collisions, and they are to 90% elastic to 10% plastic. Okay, and at the end of the simulation, we get configurations like this one. And now we are then trying to analyze these configurations. So basically what we see here as physicists is a network of droplets. And first of all, of course, we define an adjacency matrix between pairs of droplets. So the value of matrix eta of ij is equal to 1 if the droplet I and the droplet j touch each other or even slightly overlap with each other, and 0 otherwise. And now we can, of course, perform some network analysis, for which there are standardly 4 different levels. We can have a look at the atomic level, in which we consider only single nodes, at a group level, in which we consider a group of nodes, or at a global level, for example, if we have a look at the number of clusters and the maximum cluster size, or whether the overall network is percolating. And also we can have a look at some local global point of view, for example, if some single node is very important for the overall network. Here I first of all give you some example for the standard network analysis. Here, for example, you see the sigmoidal increase of the average number of degrees of a particle. So the degree is defined as the number of particles one particle is attached to. And you see how this average number here increases over time for various system sizes. For 5,000, 4,000, 3,000, 2,000, and 1,000, we get a very significant sigmoidal decrease. But for small system sizes, we see, of course, that the average number also is much smaller at the end of the simulation, meaning that there are only some isolated particles lying at the bottom of the cylinder. They don't form a large network anymore. We can also have a closer look at the final average degree of particles for various system sizes. And if we analyze them closely, we see that we get really interesting results. We get a critical behavior. We find formulas like, for example, a quadratic increase for very small system sizes, a critical increase for medium system sizes, and also what is an increase rather similar to the one in the Ising model for very large system sizes. And I have also added some further results for numbers of classes and maximum cluster sizes in the conference paper. But within this work, we are mainly interested in another question. First of all, if I choose an arbitrary pair of droplets, then the question is there a path between this pair of droplets? And secondly, how long is this path on average? So we start first of all off with counting the number NP of paths. Of course, this number is restricted. This is larger or equal to 0. And of course, it is smaller or equal to the number in which there is a path from any droplet to any other droplet. This is, of course, the maximum n times n minus 1 divided by 2. Of course, then we can define a fraction, fp of existing paths, which is a number which is limited between 0 and 1. This is then normalized. Okay, and secondly, we have a look at the geodesic distance of a pair of nodes, which is simply defined as the minimum number of edges to be transferred to get from node I to node J. So here are some results. First of all, for the fraction of paths, again, you see a nice sigmoidal increase over time. The insert shows you that it is indeed a double sigmoidal increase. So first one at very short time scales, and then a second one at medium time scales. But even more important and more interesting are the results for the average geodesic distance. Here we see the time evolution of this average, and we see especially for the largest system sizes that there is an intermediate maximum of this average geodesic distance. And this value is much larger than the final values which are reached at the end of the simulation. So we wonder what is going on here. So first of all, we have a look at the time in which this intermediate maximum occurs for various system sizes. And we see that this time depends on the system size itself. It is the smaller, the larger the system size is, and it scales with 1 over the system size, 1 over the number of droplets. Then we also have again a look at the final value, at the final average geodesic distance for even more system sizes, which were not shown in the previous slide. And you see here that we get also such an intermediate maximum at system size at roughly 700 nodes, which means that here with the final values, we somehow reflect what is happening during a simulation of much larger system sizes. Okay, so obviously, this maximum geodesic distance is not found for large system sizes in which we have truly three-dimensional agglomerations of droplets at the bottom of a cylinder, but the maximum is found in for cases in which we find a fractal structure, which is, according to our first investigations, not truly two-dimensional, in which we find a fractal structure at the bottom of a cylinder. And we will continue to investigate these structures. Thank you for your attention. And finally, I want to acknowledge that this project has been supported by the European Union. And thank you for listening. Do you have any questions?
Great, thank you so much. So again, if you do have questions, you can post them in the chat or raise your hand and we can have you ask them. While we wait for questions, I have one, which is mostly so we saw a lot of results that were in simulation. What are the, I guess, real world applications of these kinds of studies?
Well, we are still examining the pictures of experiments. And then the next step, of course, is also to see how the properties of the networks created in the experiments are in accordance with the properties of the networks created in the simulations. But this is still ongoing work. We are on it.
Okay, cool. Any other questions? Okay, well, if there are any other questions, you can go ahead and again ask them on Slack if anything comes to you later. We're running about 8 minutes ahead of schedule right now. I don't want to start the next presentation too early, just in case anyone wanted to come in for the talk. So let's go ahead and take just an 8-minute break. And again, our next talk is scheduled to start on the hour. So maybe, I don't know, how to have a stretch. That's always good when we're sitting down for so long, and then come back on the hour.
Sorry, I just stepped out for a second. Are we missing a speaker?
No, the last presentation was very short, so I'm
okay,
just waiting.
So we have a little break.
I'm just having a stretch break.
All right, perfect.
Hi.
Hi there. Okay, would you like to go ahead and get your slides shared?
Do you hear me?
Yes.
Okay, perfect. Most multicellular organisms begin their life as a single cell whose progeny reliably self-assemble into highly complex anatomies with many organs and tissues in precisely the same arrangement every time. Each cell can only interact with their neighboring environment through its cell membrane, and based on the signal that it receives from the neighbors and its internal state, is able to understand what kind of cells to be and self-regulate. Despite the limited information at the disposal of every single cell, incredibly complex organisms are formed. This ability is the most fundamental skill every multicellular creature possesses, and it's arguably the most striking example of self-organization. In 1951, when he was working in self-replication, John von Neumann invented the model called cellular automaton to try to provide a theory on biological development, and it has proven over the time to be one of the best models of life. But what is a cellular automaton? A cellular automaton is a grid of cells, each of which has an internal state. The only requirement is that the next state of each cell depends only on its previous state and the state of its neighbors. This is analogous of what real cells do. Von Neumann automaton was capable of self-replication, but used 29 different states and a rather complicated dynamics. A simpler and more popular cellular automaton called the Game of Life was invented by Conway in 1980, and this is what happens if you let it run. The output of the Game of Life resembles what you will see if you look at some microbes through a microscope, hence the name.
Cells in biological organisms follow rules which are encoded in their DNA. Unfortunately, these rules cannot be modeled by simple mathematical functions. For this reason, scientists made a more refined version of the cellular automata model discussed before, which is called neural cellular automata. A neural cellular automata, or NCA for short, starts from a single seed cell, which then expands and evolves into the final shape, in this case a lizard. In this model, the state of each cell is represented by a vector with 16 components. The first 4 components represent the RGB channels of the image, and the last 12 represent hidden values that are not visible by the naked eye. These values can represent nutrients, hormone concentrations, or chemical potentials, and are used by the cellular automaton to pass information between its cells. The alpha channel is the transparency of each pixel, and it has a special meaning. It tells us if a cell is mature, growing, or empty. Cells having alpha greater than 0.1 are considered mature. Their neighbors are considered growing, and all the other cells are considered empty. Empty cells have the state vector equal to 0. We say that mature and growing cells are alive because it is where the CA rule is applied. The update rule of NCA is modeled with a neural network. This allows us to learn its parameters with gradient-based optimization by comparing the final shape that the NCA produced with the target shape that we want to reach and backpropagating the error. This way, we can train the NCA to reach the target image after some steps. We can view the parameters of the update rule in NCAs to have the same role that the DNA has in real-life organisms, which is what differentiates one organism from the other. The output of the update rule, on the other hand, is analogous to the expression of the genome, which is the image that the NCA produces. Because of these remarkable similarities between NCA and real-life organisms, this model is able to reproduce many of the properties that we see in nature. For example, we can train an NCA to be able to regenerate a missing limb, which is similar to what salamanders manage to do in real life. Another interesting property of NCA is that they have a mechanism similar to aging. In fact, depending on how you train the NCA, you get different results. For example, growing NCAs grow into a final state and then decay. This represents growing and aging biological organisms. These NCAs can be considered mortal because after some time, the system decays. There are also persistent NCAs, which grow into a final state and keep it for an infinite amount of time. This is equivalent to an non-aging organism, like, for example, lobsters, which have this property.
Since NCAs are a good representation of living organisms, we can assume that if something works in an NCA, then chances are it will work on living organisms as well. So we can try to answer the question. Given a starting rule, can we change its properties and behavior? For example, changing the color or the shape or even both? Let's say we want to modify the behavior of an already grown organism by adding some new cells with slightly different weights, which, as said before, is equivalent to adding cells with slightly different DNA and see if they are able to steer the properties of the entire organism. And here are the results we obtained. As you can see, the cells learn to take over the entire organism to be able to accomplish their goal. In this case, the purple cells represent the new ones, while the orange cells, the old ones. We can see that the purple cells expand in space and take over the old cells over time. These adversaries can be programmed to perform a different task, for example, changing the color of the organism. To be able to accomplish this, we had to generalize the model from Morditsev and al. Before, a cell state was represented by a state vector having the first 4 components representing the RGBA of the pixel. And the remaining were hidden channels that helped the CA pass information between its cells. If the alpha channel was bigger than 0.1, it means that the cell is alive, otherwise it's dead. If we are going to have 2 different cell types, we are going to need 2 different alpha channels, one for each cell type. Since a cell cannot be of both kinds at the same time, we chose that if alpha 1, which is the alpha value of the first kind of cell, is bigger than 0.1, then alpha 2 must be 0, and the cell update follows F1, and vice versa. So a cell can be alive in only one channel. You can have a live cell that behaves like both. This is because if we interpret the 2 cells as having different DNA, then they must have different rule and there is no in between. We also impose that a new cell can only grow near a mature cell of the same type. For example, cells of type 2 can only grow near mature cells of type 2, unless the space is already occupied by another mature cell. If we do the same reasoning for the orange cells, we get this. If you look closely, there are some magenta squares. They are the combination of light purple and light orange. These squares neighbor both kinds of mature cells. These cells have both alpha channels below 0.1 and follow an average of both rules. Keep in mind that neither up-to-date rules can directly influence their neighboring cells. So the adversarial is forced to rely on changing its internal state in such a way that the original cells go through a process of apoptosis. Also, to make the job harder for the adversaries, we did so that they can't trivially discern the kind of an egg-boring cell. This method works so well that we managed to make it work even by injecting one single cell into the system. Now let's take a closer look on how the adversarial cells take over the original cells and change the organism. In this example, we start by injecting the adversaries in a 2x2 square. The adversaries can't act directly on the neighboring cells, so they change their internal state in order to trigger the process of apoptosis in the original cells. Once the alpha channel of the neighboring cell goes below 0.1, a new adversarial can go in that place and replace the old one. And the process goes on until all the cells are replaced with adversaries. In practice, however, these steps are much quicker and merged together.
We also trained adversaries to edit organisms in many other ways, and they all are oddly satisfying to watch. For example, we trained adversaries that cut off the tail of the lizard, or even turn a bug into a butterfly. Note that in all of these examples, the starting position of the adversaries is always random, and the initial percentage of adversaries is less than 1% of the original cells. We also performed other experiments, like stopping the aging of a growing NCA. For example, starting from this growing lizard, we trained adversaries to stop the aging of the organism. In this case, the adversaries must perform the takeover before the organism decays. So sometimes a higher percentage of adversaries will be needed. This is the case of the firework, which decays much quicker. As a consequence, we need a higher percentage of adversaries. The main similarities between neural cellular automata and real biological organisms may indicate that a more refined technique could be used to bioengineer already living organisms. For example, in the future, we might be able to inject agents in our body that will restore a missing limb, or add a completely new one if you really want to. Other possibilities might be to stop aging, improve organ function, or cognitive capabilities. To be able to reach this goal, however, the main challenge is to understand how the DNA and pharmacological reagents act at the low level. Once this is done, we can develop a computational model that emulates low-level cell behavior. With this program, we'll be able to input the desired changes at the high level, and the program will calculate the low-level parameters to tune in order to reach the objective. And once this is done, we can apply it to the real organism. There has been some research that shows us that the information to perform tasks like stopping aging or regenerate major damage is already present in the organism. We only need to trigger the process. Adversaries, however, can also be used in this case. For example, we can inject adversaries near a missing limb, and they will trigger the regeneration process and finally go away, leaving the space to the original cells. Or even better, we can create a sort of vaccine against damage, making the organism permanently able to regenerate. The possibilities are endless, and this could lead to exciting new work in medicine and computer science down the road. We hope that you enjoyed the talk. We also created a collab notebook where you can train your own adversarial model and play with the experiments. All the information are available by visiting this website, bit.ly/ALifeGoku, or by scanning the QR code. Finally, me and Francesco are currently looking for a job or PhD position. So if you are interested, please contact us.
Okay, great. Thank you. Do we have any questions? Cool. There's a question from Hiroki in the chat. It says, very impressive work. Can the initial adversarial cell be generated autonomously and endogenously once the organism reaches a certain developmental stage?
Well, usually these cells are produced in a way that the algorithm learns to create a cell that is able to satisfy an objective. So it is not something that happens spontaneously. There has to be an external agent that wants to modify something about the organism and then apply it to the organism. I hope this answers the question.
Okay, cool. Any other questions? So I guess my question about this kind of work is so at the end of your talk, you mentioned a lot of sort of cool potential future applications with like regenerative medicine and things like that. What would be the next step of research that needs to be done to sort of bridge that gap between doing the image-based stuff and going to like actual regenerative medicine?
Yeah, more the next step, there are a lot of steps and all of them are pretty hard. And the main thing is to be able to create a simulation that once you have a sequence of DNA, you are able to understand what kind of organism grows from their embryo. So for example, nowadays we might know that some genes are related to blue eyes, but we do this by association, not because we can simulate it and understand how it works at a low level. So understanding how it works at a low level, the DNA will empower us to engineer this kind of simulations.
Okay, cool.
Thanks.
We have a comment from Lana.
Yes, I believe that this kind of relations, I mean, all we did in this work in the end was to try to model how different kind of cells interact with each other. And this is something you see in nature, in parasite, but also in fungus on trees. So yes, there are some similarities with what happens in nature. It's a perfect example.
Okay.
The images. Well, in theory, there is nothing stopping us to do that. The problem that might become a little bit harder would be to generate a target image in 3D. So you have to make like a target sculpture with also the inside cells. You know, like a Minecraft statue. It can be done, but I mean, one of there is also, no, I stop here. It's better to stop here. I hope this is clear. I was clear to answer.
All right, well, thanks again for your talk. Maybe we'll go ahead and have the next speaker come up, and I'm going to hand the reins over to Olaf for chairing.
Thanks.
Thanks a lot, Lisa. Thanks everyone so far. I just joined recently this session, but I think the next talk is by Piper in the paper by Chiara Johnson et al. And yeah, Piper, feel free to share your slides.
I'll be
already.
sharing the video for them actually.
Oh, all right, yeah. That worked as well. Sorry, I'm looking at the short version where I didn't put your name.
Yeah. I have the better internet connection. So.
That's what's happening. All right. Okay, and let's wait for 30 seconds so that we hit time. Oh, and who is going to be presenting? Anya, is it you or is it Chiara or?
I believe it's a video, no?
Oh, it's a video?
Yeah, it's a video.
Right.
I was waiting to start the share until the 30 seconds, but I can start whatever you want.
Yeah, let's wait for a few more seconds.
Yeah.
And yeah, let's go.
I just realized I forgot to click the check boxes that need to be clicked. So give me 1 more second.
Let's check the boxes.
Yes.
I like to say very useful things, as you can see.
Okay, so now the sound should work.
Awesome.
Hello, and welcome to our talk on in vitro modeling of endosymbiotic evolution. I'm Chiara Johnson, this is Piper Welch, and this research was conducted in collaboration with Dr. Anya Vostenar from Carleton College and Dr. Emily Dolson from Michigan State University.
So I'll start off by going into the background of the relevant biological information. Our research focuses on symbiosis, which is a close and sustained relationship between a host and symbiont. Specifically, we're focusing on two subtypes of symbiosis, endosymbiosis and ectosymbiosis. Endosymbiosis occurs when a symbiont lives inside or within a host. Some common examples of this are the human gut microbiome and the mitochondria. Ectosymbiosis, on the other hand, occurs when a symbiont lives on the host. Common examples of this include ticks, fleas, and clownfish. Another important aspect of the relevant biology is parasitism and mutualism. All individuals in a symbiotic relationship exist on the spectrum from parasitic to mutualistic. Mutualistic symbionts will confer some benefit onto their partner, while antagonistic symbionts will cause some sort of harm to their partner, typically in the form of resource siphoning. Now I'll be going into the relevance of this research. Endosymbiosis has played a crucial role in the development of complex multicellular eukaryotic life. Three important endosymbionts are one, plastids, which allowed for photosynthesis to develop as a way of harvesting light energy and turning it into glucose. Two, the mitochondria, that has provided eukaryotic cells with the chemical energy that allowed for the rapid expansion and diversification of the clade. And finally, the gut microbiome, that has an integral role in supporting human health, such as aiding in digestion, vitamin K production, and immune defense. So clearly, endosymbiosis has a very important role in our existence and lives. So uncovering the possible evolutionary pathways that endosymbionts have taken is important to better understanding the human system.
There exist two proposed hypotheses for the evolutionary paths of endosymbiosis, the Black Queen hypothesis and the co-opted antagonist hypothesis. The Black Queen hypothesis posits that obligate endosymbiosis may evolve from an existing facultative mutualism, which becomes obligate when one or more partners lose the ability to survive independently, so they start out in a relationship and then they come to depend upon each other. The co-opted antagonist hypothesis proposes that an initially antagonistic relationship evolves into mutualism. Our research questions were: under what conditions does endosymbiosis evolve, and which hypothesis does the evolution of endosymbiosis follow?
To actually conduct our investigation on the de novo evolution of obligate endosymbiosis, we use simulation, an open source agent-based modeling platform for the study of endosymbiosis. In simulation, organisms that successfully collect resources are allowed to reproduce and pass on their genome with a potentially mutated value. Two things that simulation affords is the necessary elements for evolution via natural selection, which are time, variation, competition, and inheritance, both for a population of hosts and symbionts, and the possibility of an interaction between an individual host and a symbiont that is anywhere along the spectrum between mutualism and parasitism.
Hosts and symbionts live in functionally distinct worlds from each other, and the two species do not compete with each other for space. Initially, there is no interaction between hosts and symbionts. Over the course of the experiment, symbionts can mutate towards infecting and interacting with hosts, developing endosymbiosis. Symbiotic behavior and independent fitness are inversely related. The more strongly organisms evolve to interact with one another, the fewer resources they receive when they don't engage in symbiosis. Every update, each organism gets a set number of the resources from the world. Depending upon how much they've evolved to interact, they may put those resources towards their own reproduction or spend some number of them trying to interact. Thus, when they do not have a partner but try to interact regardless, they are wasting resources. When they pass a certain resource threshold, they reproduce and pass on their interaction value, which may mutate. We have an intermediate interaction state of ectosymbiosis between endosymbiosis and living independently. If ectosymbiosis is permitted in the world, a symbiont and nearby hosts can exchange or steal resources without the symbiont committing fully to the host, as it does when it engages in endosymbiosis and infects. Before exploring how endosymbiosis and ectosymbiosis combine to influence the evolution of obligate symbiosis, we considered how they evolved in isolation. First, we inspected how ectosymbiosis in isolation might affect symbiotic evolution. On the x-axis, we have how many resources a host receives per update. Colors indicate how many resources free living, not endosymbiotic, symbionts receive per update. And on the y-axis, we have average symbiont behavior, ranging from -1 parasitic to 0 neutral to 1 mutualistic. We observe that when hosts receive very few resources and it is not worth it for the symbionts to trade off their own independent resource collecting abilities in order to get resources from hosts. As hosts gain more resources, however, symbionts evolve more symbiotic behavior, particularly in conditions where symbionts are not very independently fit. What we don't see in these conditions, however, is the evolution of meaningful mutualism, potentially due to the lack of sustained relationship between one host and one symbiont. Symbionts evolve to be more exploitative than beneficial. When only ectosymbiosis is present in this system, we observe a minimum threshold of resources required for organisms to survive and evolve interactive behaviors, and mutualism does not usually evolve. Second, we examined how exclusively endosymbiosis may affect the evolution of cooperation. Here we look at the ratio of hosted to free symbionts in the world, the number of symbionts inside hosts versus the number of symbionts outside a host. When symbionts receive 10 resources per update from the world, they do not become endosymbiotic. More generally, when organisms receive many or few resources, we see fewer endosymbionts as a proportion of total symbionts. Instead, endosymbiosis is most prevalent at intermediary resource levels. They have enough resources to reproduce and evolve, but not so many that independence is more worthwhile than symbiosis. Looking at the interaction values for endosymbionts, we observe that meaningful symbiotic behavior was more likely to evolve when hosts received many resources from the world. Additionally, when symbionts received either few or many resources from the world, they would often evolve to be parasitic. However, when hosts received intermediate resources from the world, then as symbionts received more resources, they were more likely to evolve towards mutualism. Altogether, the number of resources organisms received from the world significantly affected the evolution of behavior, and there were a narrow range of resource levels that led to mutualism when only endosymbiosis was permitted. Third, we explored how ectosymbiosis and endosymbiosis could combine to affect evolutionary trajectories. Comparing experiments run under the same condition, one with only endosymbiosis and one with both endosymbiosis and ectosymbiosis, we found that the presence of ectosymbiosis decreased the evolution of endosymbiosis in some conditions, such as for intermediate resource values. However, in conditions unideal for endosymbiosis, ectosymbiosis encouraged the evolution of obligate symbiosis. These included cases such as when symbionts received many resources from the world and were independently fit, thus disinclined to fully commit to interacting with hosts, or situations where symbionts received very few resources from the world and thus struggled to evolve symbiotic behavior independently. These results suggest that when ectosymbiosis is permitted, endosymbiosis can evolve in a more diverse set of conditions, but may evolve less strongly in ideal conditions. Finally, we examined the trajectories for the evolution of endosymbiosis under these conditions. We tracked the phylogenies for the clones that dominated the worlds at the end of the run. When ectosymbiosis was permitted, fewer lineages spent time in the most parasitic state, and lineages evolved towards mutualism more rapidly. These results largely support the co-opted antagonist hypothesis, as we often see symbionts engage in parasitic behavior before they eventually become mutualistic. When ectosymbiosis is present, the Black Queen hypothesis is more possible, perhaps because there is less risk involved in evolving mutualism. Their offspring are not tied to their symbiotic partner the way that in some endosymbiosis they might be. Generally, in conditions where mutualism is likely to evolve, ectosymbiosis hastens that evolution and increases its probability.
So here's some future work that we're interested in conducting. In general, we are interested in how more complex environments influence the evolution of endosymbiosis. Specifically, we're interested in how spatial structure, the option of multiple symbionts per host, and different resource types would influence the evolution of endosymbiosis. We're also interested in conducting a more in-depth investigation of the level of resources that favors endosymbiosis. Thanks for listening. We want to give a special thanks to NSF grant number 1750125 and Carleton College's Towsley Endowment. And are there any questions?
Wow, amazing. Very nice short talk. So we have plenty of time for questions. Yeah, does anyone want to start off? Small clarification, you said at some point meaningful mutualism. And what did you mean by that? I know it's a detail, but...
Kai, do you want to answer?
Hello. Meaningful mutualism, I mean, particularly because the more mutualistic in our system they get, the less independently fit they are. It's possible for them to be slightly mutualistic at 0.1 or 0.2, but still be possible for them to survive independently very strongly. And so when we talk about meaningful mutualism, we're thinking like 0.7, very high mutualistic values that essentially makes it impossible for them to live without a partner.
I see. Interesting. Very nice. So there's a sort of threshold that you have. Yeah, good. Yeah, any comments, questions in the room? Maybe I give you some space. So where do you want to bring that study next? You cover a lot of things. I think it's maybe I was personally a bit lost, but maybe I didn't focus enough. So where do you want to take that next, and what big questions do you think you will address with this model?
I think we're just interested in more complex environments, like further replicate how and like our common endless environments might evolve. So things like different resource types, like there is in the real world, like different molecules, like a glucose resource or like a carbon, things like that.
Do you think? Okay, so the billion-dollar question is how difficult is it to apply this to real nature? So the difference between the in silico that you have here and the reality. Have you done? So I haven't looked at the review that you have in the paper, but yeah, how do you address the potential publication of this in PNAS or biology journals?
Yeah, so we don't really have a wet lab operator who is able to, or a collaborator who would be able to kind of go and do this. So that's something that's a little bit further off. But there's a question in the chat.
Yeah. So Klaus, you have a question. Do you want to ask it yourself?
Sure. Hello, ALife. Hello, Piper, and everyone else. Thank you. It was a very interesting talk. I really appreciate the idea of studying the effect of environments in ecologies. And I was wondering, it was not, maybe I missed something, but if not, can you think of making this let the agents make the environment more complex? I don't know if that's if you can see that in your current ideas.
Yeah, great question. I mean, the whole question of adding complexity to an artificial model is the burning eternal question. But with environment in particular, proposed extension that we're thinking about is sort of resource values in the environment being then dictated by organisms, and then they can perhaps release resources back into the environment. And it can just be this interplay between potentially reaching some stable state versus potentially not. At this moment, however, there isn't a lot of environmental complexity, but we're definitely considering it in terms of future work. Thank you.
All right, thank you very much.
So for those who know less about symbiosis in general, what's the map looking like? So is there so there are other types of, I don't remember if you covered that in the beginning, other types of symbioses than ectosymbiosis, right? So what does it look like more? What is it free in terms of, I guess, frequency of interacting with the host, or how do you characterize it? And would the modeling become different if you address that, or would that be pretty similar with a sort of frequency added or a multiplicative term?
So like if we added other types of endosymbiosis to the model other than ecto and endo, is that what you're saying?
Yeah.
Yeah, so like we had to add the functionality to the model for ecto. So yeah, we would have to add any additional functionality and like, you know, code. But yeah, it does, like each one comes with its own like niche of, you know, properties and things. Like in like endo, then there's the issue of how many different hosts to allow in the symbionts and that kind of thing, and those kind of considerations. Or in ecto, you can have multiple symbionts per host, and those kinds of considerations come with adding any type of new symbiosis.
You allow for second order symbiosis, so some kind of species that come in there and then start creating a sort of mini ecosystem, or is that pushing it?
We do. I mean, we do have the possibility for more than one endosymbiont. The results we showed, it was just one symbiont in a host, so we didn't really get that interior niche. But it is a possibility with the model, potentially for future work. But because of vertical transmission as well, we do occasionally get like lineages that evolve to be very specific to their condition or whatever. And so we do get some sort of speciation with different behaviors depending upon particular relationships between host and symbiont and whatever behavior they've evolved.
Thanks. Yeah, any other questions? I'll have to have another look at the paper. I think there's a lot in there. Other questions, comments? All right, then yeah, I want to thank the speakers again for a very nicely prepared recording, and yeah, let's give ourselves another minute before we go to the next presentation. So maybe the next speaker, Alison, can already put your slides up, and we'll wait a minute.
Allison, just checking. Do you got it, or do you need me to?
I got it.
So, Anya, you're just on every battle, aren't you?
Yes, it's a busy week.
Good. Those are the best weeks.
Okay, actually, Anya, I'm sorry, the new laptop, I don't have all the settings ready to go. So if you wouldn't mind, that would be great.
Yep, no worries. All right, round 2 here. Let me make sure I click all buttons correctly. I'll try to actually do it here.
Hi, everybody. My name is Allison Cameron, and today I will be talking about our paper, which is titled Keep Your Frenemies Closer. Bacteriophage that benefit their hosts evolve to be more temperate.
Sorry. Yeah, please restart.
Yep, will do. Sorry, I've got like two screens and I keep losing my mouse. It's very annoying. Are we good, Olaf, or?
Yeah, sorry about that. I saw a 39 and then jumped to 40. Anyways,
Yep,
yeah, let's go.
Hi, everybody. My name is Allison Cameron, and today I will be talking about our paper, which is titled Keep Your Frenemies Closer. Bacteriophage that benefit their hosts evolve to be more temperate. A little roadmap of what the presentation is going to look like. First, a background, then a methodology. Next, I'll show you our results, and finally, a conclusion. So first, let's get into our background. Now, what are bacteriophage? Bacteriophage are viruses that specifically target bacteria. So this means that they can only infect bacteria and no other organisms. Bacteriophage, or phage, as I'll refer to them as such in this presentation, are unique in that they can choose between a number of reproductive strategies. More concretely, they can choose between lysis and lysogeny, which I'll go over more in detail in just a moment. Because of these reproductive strategies, bacteriophage have a large effect on their bacterial hosts. And so you end up with a lot of co-evolutionary dynamics, such as the Red Queen hypothesis, which describes species that push each other to continue to evolve and evolve more defense mechanisms or weapons against one another. You also end up with Lotka-Volterra dynamics, which are kind of a predator-prey population size fluctuations. And so you can see that with bacteriophage and bacteria, where phage are the predators and bacteria are the prey. Examples of where this shows up are in our human gut microbiome. So we've got millions of little bacteria inside of us, as well as bacteriophage that are influencing how those bacteria evolve. Another important point for this is that with the rise of antibiotic resistance, bacteriophage are being considered as phage therapy to actually target harmful bacteria. However, we need to understand how phage and bacteria co-evolve. We need to understand that better in order for it to actually be used in a clinical setting. So now I'll go over the different reproductive strategies now that I've gone over our motivation for this project. So first, we're going to talk about lysis. So the spidery looking thing is a phage, and obviously the oval is a bacteria. So the phage finds an uninfected bacteria, it injects its DNA into the bacteria, and now for lysis, the DNA will hijack the host metabolism and start creating viral particles. These viral particles will then eventually be assembled into actual phage offspring. And lastly, the bacterial cell will be bursted. So the host actually dies once the offspring are released into the world, and subsequently they find new bacteria to then infect. So next, we'll talk about lysogeny, where again you have a phage finding a bacteria. The phage then injects its DNA into the bacteria. The next step of this, which is distinctive from lysis, is that genetic material of the phage actually gets incorporated into the genetic material of the host cell. And so once it gets incorporated, it's known as a prophage. Then once the host starts reproducing, so it starts copying all of its genetic material, it's actually also going to copy the prophage genetic material since it's been incorporated. So when reproduction is complete, you end up with prophage also having reproduced. So you have two copies of, or you have two versions, two offspring of the bacterial cell, each with their own version of prophage in it. So we're going to look at these two reproductive strategies through the lens of symbiosis, specifically parasitism and mutualism. So parasitism, as you might know, is when one organism benefits at the detriment of another, whereas mutualism is a net benefit from both organisms. So we can consider lysis as a parasitic relationship. Because of the diagram that you saw, the host dies, the phage reproduces, whereas lysogeny could be considered as more mutualistic. Now, this is because the fitness or the reproduction of the lysogenic phage is actually tied to the fitness of the host. So there's some incentive for a lysogenic phage to improve the host fitness. So that can lead to a mutualistic relationship. However, there are some complications with this. So, as I mentioned, you could confer beneficial traits to the host through the prophage, and this has been seen through serotype conversion as well as immunity to future phage infections. However, you could also harm the host DNA by incorporating into the wrong spot or producing other effects. So this is also complicated by another level, which is known as prophage induction, which is when you have an incorporated prophage and there's a chance under stressful conditions that the prophage would actually break out of its incorporation, so out of the host DNA, and enter the lytic cycle again, which then turns it into a parasitic or harmful relationship for that host. And so we know that these two things are linked because even if you have a mutualistic relationship between host and phage, that relationship is very perilous, and you might end up with a phage that chooses to induce back into the lytic cycle, causing a harmful relationship again. So we came up with our research questions. First, when should temperate phage lysogenize if they're able to impact host fitness? Next, does more frequent lysogeny select for beneficial prophage? And lastly, how do these phenomena reinforce each other? So now I'll get into our methodology a little bit. So we used simulation, which is an agent-based model that was developed by Dr. Anya Vostanar, who is a collaborator on this project. And in this model, there's a world with various constraints, as well as host and symbiont organisms. In this diagram, the hosts are the squares and the symbionts are the circles. The world distributes resources to each of the hosts. The hosts then use those resources to interact with their symbionts. The symbionts can take resources or add to them, multiply them, and then once each organism reaches a specific threshold of resources, they're permitted to reproduce. So the hosts and symbionts that effectively gather resources the quickest are going to be able to reproduce the fastest and therefore dominate the population. So through this model, we can observe evolution. We extended simulation to create a phage mode. So we created a subclass of a host, our bacterium, and we added just one genetic value to that, and that's the incorporation value, which I'll go over more in detail later. Then we took the symbiont and we extended a phage class from there. And our phage class has a number of characteristics. The first is whether or not they are free living or inside of the host. So our phage actually have the ability to live outside the host. They can't reproduce without them though. We have the chances of lysis, so whether or not they're going to choose lysis or lysogeny, the two reproductive strategies, as well as a chance of induction, which, as I just mentioned, is when you have a lysogenic prophage that then chooses to induce back into the lytic cycle. So how frequently is that happening? And lastly, the incorporation value, which I'll go over more in a moment. So we calibrated our phage mode. This is work of Wall et al., and I'm not going to go over it in super detail, but essentially what it's trying to tell us is that when you have a limited number of hosts, you end up with more lysogeny, and when you have unlimited hosts, you end up with more lysis. This is because the lytic phage kill their hosts, so if they kill all of the limited available hosts, they will have no way to reproduce. So main takeaway, more available bacterial hosts select for higher rates of lysis, and conversely, a limited number of hosts selects for more lysogeny. So what did we do to calibrate? We implemented a prophage loss rate, which basically means at every time step, we check the prophage or the lysogenic phage if it's time for them to die. So this mimics the denigration of genetic material. And so as you can see at low prophage loss rates, so 0, this blue line here, we end up with high rates of lysogeny. So that means low rates of lysis. Conversely, at a prophage loss rate of 5%, so high prophage loss rate, we end up with much more lysis. So this means that our higher prophage loss rates are selecting for lysis, which the higher prophage loss rates means more available hosts, and conversely, no prophage loss, so a limited number of hosts, is going to select for lysogeny. Ultra main takeaway is that our system matches the results of previous work. So now let's get into our results. First, we asked what effect does prophage induction have on the lysis-lysogeny decision? So as I mentioned, we implemented induction as just a small chance that prophage induced back to the lytic cycle. So on the left-hand side, we have our naive experiment, which is pre the implementation of induction. And on the right-hand side, we have varying starting induction chances, which were allowed to mutate throughout the experiment. So as we can see, the chance of lysis actually goes down no matter the prophage loss rate and no matter the induction chance. So we hypothesized that this is because choosing lysogeny when you have the chance of induction is a much less binding decision than when one doesn't have the chance of induction. So in conclusion, the induction into the lytic cycle does significantly affect the lysis-lysogeny decision. Next, we wanted to know what the effect of non-dormant prophage have on the lysis-lysogeny decision. So how did we implement this non-dormancy? We created what we called an incorporation value, which is basically just a number between 0 and 1 that each organism has. So when you end up with a bacterium and phage pair, and you know that this phage is lysogenic, then what they're going to do is compare these values. So the values, depending on how similar they are, can range from canceling out all new resources for the host to doubling them. So the closer that incorporation value is, the more helpful the phage will be to the host. So in this panel here on the very left-hand side, you can see that the incorporation values are very similar. So you have a high number of resources here. Conversely, in this situation where the incorporation values are very different, you have a very low number of resources. And that's what this heat map is again trying to reinforce, is that when they're very different, you end up with 0 resources, whereas when they're very similar, you end up with 100. So this is in the situation where they're receiving 50 resources from the world. So again, either canceling out or doubling those resources. What did we learn? So on the right-hand or left-hand side is our dormant prophage, our naive experiment. And on the left-hand side are 3 columns of non-dormancy with different starting populations. So this figure is showing us that when you have harmful phage, you end up with much higher rates of lysis. So this reinforces our hypotheses and our way of looking at it through the lens of symbiosis, that the harmful or parasitic relationship is going to end up with more lysis. Conversely, with beneficial phage, you end up with more lysogeny, which is kind of more of a mutualistic relationship as long as they're still beneficial. So we learned that even in situations where typically you would expect dormant prophage to be lytic, when you have beneficial phage, so that non-dormancy, they end up being much more lysogenic. So our main takeaway is that the prophage impact on host resources does significantly affect the lysis-lysogeny decision. More specifically, beneficial phage tend strongly towards lysogeny. So in conclusion, we know that this is really important because the prophage are affecting the evolution, or these different factors are affecting the evolution of the phage reproductive strategy. So the ability for prophage to benefit their hosts has a substantial impact on the evolution of the phage reproductive strategy, as we've shown in our results sections. So because the reproductive strategy is so important for how it impacts the hosts, the evolution of the phage reproductive strategy then impacts the evolution of the bacterial hosts. So this has important implications for understanding microbiome makeups. So these coevolutionary dynamics are pervasive, and our work has shown this. And therefore, it's really important to our understanding of microbiomes to understand phage-host coevolution dynamics. And then once we understand microbiomes and how phage and hosts are affecting microbiomes, we can understand their effects on larger ecosystems. So this non-dormancy, which has only recently been considered to be important, might have really significant effects on the way that we view our world in an ecological sense. So future work, obviously there's a lot more to do in this area. So firstly, we want to look at the combined effects of induction and non-dormancy. We looked at them isolated here, so seeing how they interact could be interesting. Additionally, we would want to look at spatial structure. So these experiments were conducted in a well-mixed or aqueous environment, which means that when an organism reproduces, their offspring is just placed into a random spot in the world. So we think that adding spatial structure might allow us to see more pockets of traits or ecological niches, which could be interesting. We also want to explore a higher multiplicity of infection. In this work, we only allowed for each bacterium to be infected by a singular phage. In reality, that's generally not the case. Bacteriums can be infected by multiple phage, and those phage can then interact with each other once they're inside the host cell. So we'd like to allow that to happen as well. Of course, we also want to test these results in the wet lab and verify them and make sure what we're seeing is actually what happens in real life. And that's important for understanding of phage therapy and medical relevance, and in order to develop an effective response to antibiotic resistance. So thank you so much for listening to our presentation. This project would not have been possible if it weren't for our funders, as well as all of my collaborators. So thank you to all of the people that supported this project along the way. I'll now open it up for questions. I've left a methods figure, a results figure, as well as QR codes to our source code and the paper preprint. So if you're interested, scan it, take a look, let me know what you think, and thank you so much for listening.
Amazing. Thank you, Alison, and co-authors, I guess. Yeah, amazing presentation again. Yeah, it was just everything very clear and neat and on time. This is the future. All right, any questions from the room? Audience, any comment or question? You can raise your hand or post it in the chat, either way. Yeah, Klaus.
Hello, Alison. Thank you very much for the talk. Very interesting. I want to ask like a very, very trivial question. I'm not a wet person at all. So when you go in the end and you say like, okay, we want to reproduce these in the wet lab at the end to see to validate. If I understood correctly, you're doing like you're varying the capacity of the microbes to have this phage relation. And I mean, you do a lot of manipulations of what the microbes can or cannot do. And is that easy to manipulate that in the wet lab? How does that happen? I mean, is that something that is really like, is it like you said, okay, we're just going to reproduce these on the web? And my experience is like usually it's web to digital, not digital to lab. So can you talk a little bit about that?
Yeah, sure. So part of the reason that we use simulation is to kind of overcome those constraints of what you can do in a wet lab. So that's both like the expense of doing these things as well as like time constraints, because it takes a long time to observe evolution. So it's definitely not as easy. So that's why it's like beneficial to start with doing a model of it and then see what comes out of it. And maybe you can try to do something like that. I don't have a super great understanding of how exactly you would do this in a wet lab, but you know, there are ways to edit the genes of various organisms. So perhaps you edit a gene of a phage so that it can do serotype conversion on its host, which is basically that it changes the protein structure on the outside of the cell so that the cell can bind to other cells or like other phages easier or worse. So you could do something like that in terms of providing a benefit, as well as like certain phage can provide immunity to future phage infections. So I think there are ways where you can kind of edit the genes to do that kind of stuff, but I'm not a biology expert, so that's just my high level understanding. But thank you for the question. Hopefully that answered.
All right. Yeah, I think it gave me an idea. Although, yeah, I think as we're talking like non-biologists to each other. So, okay, cool. Thank you.
So, I mean, I'll jump in quick. One of our co-authors, Sarah Dorr, is a virologist, right? She's our resident person, and she is working on. She has some really great lines of a bacterium called Shigella, which has a number of different phage that we suspect do have these sorts of relationships. And so, yeah, we are gearing up once she's able to get her wet lab evolution experiments going to be able to do some longer term evolution experiments to see what impact these different phage have and kind of what they evolve to do. So probably not next year, but maybe the year after.
Cool. Sounds exciting. Thank you.
Right. Yeah, thank you so much again. I just quick question. There is no spatial simulation of this, right? It's only like coefficients and chances of induction and so on. Is that correct?
Yeah, Anya, you'll have to help me with what the shape of the world is, but there's like a little slot for each organism. So it's a vector, but it's not quite just like a 2D array. I forget what the shape is called. But it's the point of that it being well mixed is just that the offspring are placed into random spots into this world.
I see.
So they're like kind of is a spatial structure, but we didn't take advantage of it.
Got it. Yeah.
Yeah,
Right.
we can turn on toroidal, like, the donut shape if in the future.
I see. So it's a torus, but yeah, you can enrich it, I guess. Yeah, that would be interesting. Yeah. All right. Yeah. Thank you again. Very nice talk. And our next one up is by Tyrell Davis. Feel free to share your slides. And it's another 15 plus 5 minutes. Questions and answers right after. I think you're muted, Tyrell. I can't hear you. Can you check your mic?
We've had this earlier. It helped to leave and rejoin back then.
All right. So the advice seems to be turn it off and on again. I mean, Zoom,
Yeah.
go out of Zoom and reconnect.
Ironically, that's always the actual fix that works.
Yeah.
Those tech people, they know what they're doing. When they
That's right.
Did you try to turn it off and on again?
I think that's the correct way. Oh, I see. There's in the meantime, he posted it only to me, but I guess I'll post it here just in case that's helpful.
It's crossed.
I see. That's for the slides. Yeah, good.
How about now?
We'll
Yes, we hear you.
We can hear you now. Perfect. Yeah, whenever you're ready.
Okay, so thanks. I also put the slides in the chat so you can follow along if you want to go back at a different pace or something like that. All right, thank you, Olaf, for introducing me. I'm going to be talking about continuous cellular automata. It's a variant of Lenia, which most of you have probably heard of. And the motivation for this is I noticed that there are certain types of life-like cellular automata, so the same family as John Conway's game of life, that don't implement very well in Lenia. In fact, it's not, there's no facile way to express them with the Lenia update function. So my name is Tyrell Davis. My PI is Josh Bongard, and I'll just get going. So this is an example of the sort of failure to express an interesting type of pattern in one of these CA. This should be moving, and the time lapse on the left is in Lenia. It becomes a still life, and on the right, we rescue the behavior with this new variation of Lenia called collaboration. Okay, so I'd like to start out with the conclusions. The first one is just that the framework, Lenia, it does not have easy implementations for lifelike CAs if those CA have distinct birth and survival rules. So by distinct, I mean non-overlapping birth and survival rules. So Conway's life works fine, and I'll talk about why in a minute. But other ones that are, and there are plenty of interesting ones that fit this profile, don't easily implement. Okay, so we can rescue this ability if we split the growth function used by Lenia into two separate functions called genesis and persistence. So those are analogs to birth and survival. And there are a few interesting CA. I'm going to focus on Morley in this talk. So that's B368 and S245. So that's birth conditions are 3, 6, and 8 neighbors. Survival conditions 2, 4, and 5 neighbors. And there's another one that I'm not going to talk about, but it's found to have optimal complexity according to a density complexity metric from this paper here from Peña and Sayama. And that one's called Dynamic Life. Okay, so as far as comparing the two frameworks, most of the dynamics for Glabrish CA are a little bit more active than most of the rule sets for Lenia. But I suspect that has more to do with how I chose the rules, and I'll talk about that later, as compared to Lenia. Okay, and this is an example of the first row is the Lenia CA hydrogeminium natans. And on the right column, that's the spatial entropy corresponding to the CA states on the left. The second row is one of these new CA, and this one's called S613. That's just because the random seed was 613 for that experiment. Okay, so I know that some of you will listen to this and say, why make something more complicated if you already have something complex enough to be computationally universal? And that's a fair criticism. In fact, we can apply it to all the extensions of CA more complicated than something like Rule 110. And you could implement these CA that I'm going to show in Linea. At least you could build a metapixel in Life implemented in Linea, and then run those other rules on the metapixel within Life, within Linea, sort of a shell game all the way down. It would take a very long time. Thousands of steps are required for each step with these metacells and so on. But you could do it. So both of these CA frameworks support universal CA. Okay, so simpler is better, but if there's something worthwhile, it can justify making things more complicated. Okay, so one thing that I'm really interested in and I like to think about, that seems like it might be a good justification for building more complicated CA, including continuous cellular automata like Linea and Glabrish, is the possibility of making agents that are living in a physically consistent universe. And what I mean by that is, in a reinforcement learning paradigm, usually the environment and the agent or the control policy are separate. In one part, you've got a physics simulation, and in another part, you've got PyTorch running neural networks or something like that. But in these continuous CA, they're a bit more complicated. But we're not just filling the computational resources we have. There's a possibility to have agents that interact with their environment. They all run under the same rules, just like we do. Just like our neurons follow the same physics as a bouncing ball and so on. We have that opportunity to do something similar in continuous CA. And this is an example of early work. And as far as I know, it's still a blog post. If anyone knows where they're going to publish this, I'd be interested in knowing. This is from Haman and others at INRIA. And they actually use backpropagation to train some rules that give rise to these gliders that can interact with these immutable objects in the environment. Okay, so the rest of the talk, I'm going to go over why implementing Life in Linea is easy, but morally in other CAs that have non-overlapping rules is not easy. And then I'm going to show how to rescue that by splitting the update function. And then I'll show some comparisons of a CA from Linea with one of the evolved CA from Glabrish. Okay, so so briefly, just the cellular automata it's just dynamic systems. They change due to rules applied in a local context. Often this is spread out on a two-dimensional grid, but you can wrap them around whatever you want and add dimensions and so on. So a typical way to simulate the dynamics of a CA is you compute the neighborhood and then you compute the update. So 2 steps. So here, the first figure here, we have the Moore neighborhood in purple of a cell in a sort of salmon color. And the next state of this cell depends on its neighbors and its current state. And that looks like something like this. So on the left, we have the cell states for a glider in Morley. And then on the right, we have the neighborhood values for each cell. And the rule for this one is B368, S245. So that means any empty cell with 3, 6, or 8 will become 1, and any non-empty cell with 2, 4, or 5 neighbors will retain its value. Okay, so Linea is similar, except the neighborhood is a smooth convolutional kernel. And the update function is also smooth. And usually the update function takes a form of a Gaussian. And in the Linea framework, it's called a growth function. And so that there's a visual of it. And if we want to look at the expression defining these dynamics, it's just right here. So it's basically Euler's numerical method for differential equations. It takes the same form. The next state is defined by the current state with the addition proportional to some step size of the result of the growth function applied to the convolution of the current state with the neighborhood kernel. And in fact, it's easy to implement Life like in this. You just make the ranges for the BNS rules and add them together. So that this is a stair-step function is in the end is what you get, with the lower step corresponding to survival and the upper step corresponding to birth, so new cells. And you can run whatever pattern you want. It should behave as expected when once you implemented life in Lenia. So this is the classic reflex glider from Conway's life. But something goes a bit wrong when you try to implement rules with non-overlapping BNS rules. So Morley has none of the rules are shared. So if you add the BNS intervals together, the best you can get is staying the same. So the values for the growth function in this case never goes above 0. And in fact, what that does is turns a dynamic puffer. This is like a glider that leaves a trail of ordered debris behind it. It becomes a still life. And this is actually an animation, which is a bit silly, but so it doesn't move, it becomes a still life. Okay, so that's the problem that I saw, and this is the solution that I ended up coming up with. So as I mentioned, it's a bit more complicated. It takes about twice as long to write out this this version. And what I did is I split the growth function into a genesis function and a persistence function. So that's the analogs to the B and the S rules. And now if we implement Morley in Glabrish, now we have distinct survival and birth conditions. And we rescue the normal behavior for that puffer. This is a pretty interesting puffer if you have time to play around with it sometimes. When I started working with Morley and just using Golly to do simulations, I would draw things that I thought might turn into gliders. And I kept on thinking I was finding new gliders, but they all were the same puffer. Okay, so how do they compare? Just by observation, most of the Lenia CA from Bert Chan's taxonomy, where he's got hundreds of patterns that have charismatic, lifelike properties. And there's an online link that you can follow later to play with them. Most of them form Turing patterns that are static if you initialize the grid with a random uniform initialization. And so that's what we see here in row B. These are 4 rules from Lenia. The top row A is evolved through random selection. So most of these fall away to a state of 0 or 1. And then the bottom 2 rows are evolved for halting, even chance of halting or persistence, or for halting unpredictability. I won't have time to talk about how they were evolved here, but you can message me later. In any case, they seem to be mostly more dynamic. You see activity continuing for a longer period of time. Okay, but that's probably because of how the rules were selected and not because of the framework, in my opinion. But we'll get to that a bit later. So, and I wanted to apply an information theoretic measure based on entropy to the to 2 specific CA from these 2 frameworks. So in this case, I do spatial entropy. So that's just entropy calculated for windows the same size as the CA neighborhood and moved across the entire grid. So first I convert to the 8-bit integer format, which makes it more computationally tractable. And then for each window, compute the entropy for that particular central location. Okay, so I'm comparing hydrogeminium natans and S613. So they both are capable of both growth and vanishing. So that meets some other criteria from Epstein's work of fertility, which is escaping from a bounding box, and mortality, which is the capability to support patterns that will just disappear after some amount of time. So those are 2 heuristics meant to suggest complexity. Both of the CA meet them. But when we look at the evolution or the progression, rather, of spatial entropy over time, we see something a little bit different. So in the left column, we have the grid states, the same starting state, a random uniform initialization in the bounding box. And on the right column, we have the spatial entropy for each grid. So C and D, that's the grid state after 512 steps for hydrogeminium natans. And E and F are the grid states and spatial entropy map for S613. So you can see that the variance in entropy over the grid is much higher after a certain number of steps has progressed for S613 as compared to the linear CA. And I'll share a link where you can go play with this yourself and see it in animation form with some notebooks online at the end. Okay, if we just plot the overall statistics for spatial entropy and how it varies over time, this is what it looks like. So in blue, we have, in the darker blue, we have the S613. So both the spatial entropy and the variance in spatial entropy, so this is plus or minus standard deviation, is higher and remains higher throughout the progression of the simulation as compared to the hydrogeminium, which, as you would expect from what you've seen in the animations, settles down to a mostly stationary state. Okay, so what are the differences between these two CA? They share the same neighborhood kernel, so that's maintained the same. And their update functions, the growth or the genesis and persistence functions, look very similar. And we can look at them here. The growth function is in almost the same place as the persistence function for this S613, but the behavior is very different. But as I mentioned, I think that it's mostly to do with how we came up with the rules rather than the framework itself. And so if we look at all of the linear CA and the patterns, I did find two that have more dynamic characteristics, Astrium scintillans and Pentifolium incarceratus, which you can check out on Chan's online demo there if you want to compare. I don't think we'll have time to do it live. And of course, this would be kind of a pointless comparison if only the linear framework, the linear CA supported gliders. But in fact, both of these CA support gliders. And the wobbly gliders are qualitatively quite similar. They have a little bit different lifetimes and different speeds and different values, but qualitatively, I could say we would say these two are related. And then we have a relatively unique frog type pattern on the right in the S613. I'm sorry, the column on the left is the linear CA hydrogeminium natans, and the S613 is on the right. And the eponymous slow, well, it's called hydrogeminium natans, the pattern as well, but I think it looks like a slow cucumber. Okay, so we made some comparisons. We did see that the S613 is a little bit more dynamic, and they both support gliders. But I think that the main differences in the absolute character is due to the way that the rules were found. So linear rules were mostly manually engineered or manually evolved by human selection, whereas I used an automated evolution process to find the particular rule parameters for the S613CA in GLabridge. Okay, so that's coming to the end of my slides, and I'll be happy to take any questions. There is also a sort of list of all the resources for this and related work. It's consolidated at this link here. And I'll be talking on Friday about some observations about how the step size parameter can have a big effect on the way that the dynamics of the CA play out. All right, so thank you very much.
Thank you so much, Taro. We have just time for one quick question, if anyone has one pressing one. Always lovely to see linear work. So Pri, I can't interrupt you.
Okay, so I see the question in the chat. Which evolution algorithm did I use to find the creatures in my rule? So to find the patterns, I used a covariance matrix adaptation evolution strategy. And what I was evolving was compositional pattern producing network parameters. So synthesis patterns that give rise to the mobile patterns. The fitness function was a combination of displacement of center of mass So just the average location of all of the cell values weighted to those cell values, and a sort of penalty for changes in the mean value of all cells. So that was a mobility and homeostasis proxies.
Thank you. All right. Well, thank you again, Tara, for your talk and beautiful images. And we'll go next to the talk by Babak. And we can see your screen. Let's see the full screen mode. But we can't hear you yet. Maybe you haven't said anything.
Yes, I lost the mute button. Sorry about that. Hello.
All right. Good.
Okay. All right. Well, let's dive right into it. This is some work that we've done at Cognizant AI Labs and UT Austin, and I'm presenting on behalf of myself and my colleagues Hormuz Shahzad and Risto Mikkulainen. And what we set out to do is basically solve problems that keep changing. Essentially, you have a problem domain that can change in its fundamentals or just changed abruptly to a different problem. And you want to be able to solve that using some sort of adaptive problem-solving system. And we can kind of cast that as a reinforcement learning problem where we have states from the problem domain coming in, actions issued, and this thing kind of loops. Problems similar to this could be, for example, playing games against an opponent that keeps changing their tactics or learning things, trading in the stock market, controlling chaotic functions. There are many sort of similar problems there. Currently, the solutions are underwhelming because they require heavy engineering. They need to be tuned to the problems usually, so they're rigid. And as the problem changes, they kind of fall apart. And typically they require a lot of training. So experiencing the problem domain a lot before you can actually let them loose on there. So the idea, the intuition was if we can actually have some form of artificial life system with many actors kind of breeding and surviving an environment, we can then have them survive and adapt to this changing problem domain. And the hope was that this system would be robust to change. The actors can improve in the abstract because they're trying to survive their local environment that is hopefully independent from the problem domain as much as possible. And we could have some transfer of knowledge. So if we have some actors that are quite specialized and as the problem domain changes, they disappear or don't survive, there are actors that do abstract out other features in the problem domain that can kind of carry over to the new problem or withstand the change. So with that in mind, what we did was we defined DS, which is the solution that we're talking about here, in a manner that is as problem independent as possible. So the way we set it up is we have an environment that in its dimensions, the dimensions are based on the dimensions of the problem that we're solving. So for example, the X dimension are the states of the problem, and the Y dimension is basically the actions that we expect from the environment. So, and then in Z, we have some actors that are kind of surviving this environment, and the actors consist of only generic states and actions, and so that provides us that separation between the state and action space of the actors and the state and action space of the problem, because these guys can only have some generic actions of moving around or checking to see what state they are in right now. And that's limited. So they don't need to expand or change their action and state space as the dimensionality of the problem changes. And this gives us a very good separation. With each iteration, the new state is kind of overlaid on the environment itself. And as the actors are surviving, they have the option to actually write a value in the location they're at. And the values within the Y dimension are averaged out as the values for the action that then is issued to the domain. They can communicate with each other indirectly through the environment, and each actor is assigned a linked location. And I'll talk briefly about what that means. But basically, what this gives us is this pressures the actors to have to communicate, to have to collaborate in order to solve the problem, because none of the actors are actually seeing the entire problem as it is. And the way we set this up is we actually defined each problem to be a rule set. So there's different types of actors that you could envision, and we did implement a bunch of different ones, from very simple random actors to like just a bandit actor that uses bandit algorithm to pick its actions, all the way to some simple reinforcement learning. Robots are just, you know, a set of rules that us programmers wrote for them, or even neuroevolution. But we settled on evolving rule sets for a number of reasons. First of all, as we're evolving the rule sets, we get a sense, explainable sense of what the behavior of each actor is, and that's helpful as we're defining and designing the system. And it worked well, so that was the other reason. And you can see here sort of a printout of one of these actors. An actor can write a 0 or 1 in its location, and that could be evolved to make use of this as a communication means of communication between actors. It is always linked to another location within the environment. And this is important because not all problems are decomposable, and we do require some sort of communication, and the actors should be able to see more than one state at a time. But we kept it at a minimal of just one linked location. And in this case, for example, the actor is checking its own domain state, which is the value of the domain state written in the location they are, to their linked location domain state. And if this condition is met, then they're writing a value. So you can see that the rules are typically very generic, and this rule could apply to any problem domain, and it would still work. And that's kind of I think the main contribution of this work. And as I said, the actors can move around. They do evolve if mates are selected from neighboring regions, and they do evolve, and that's how these rule sets are actually constructed. So you may ask, if they're evolving, how do we know how well they're working given this separation between the environment and the domain? Well, we measure what we call the contribution of an actor to the solution. So so we hopefully have a measure of the fitness of the problem, how well we're solving the problem, or at least how much better we're solving the problem itself. And if we have that, and we know whether or not an actor contributed to that solution because it actually issued or it wrote a value for an action in its location that contributed to the aggregate action that went to the domain. And if it did, then we can actually calculate that value, and we can get a sense of the contribution from this actor. Now, actors might have 3 roles that they might play. They might actually contribute to the solution. They might actually provide, write an action, but that action be ignored because the aggregation function, simply didn't pick it up. Or they may actually issue an action that has nothing to do with the domain. They might be moving around or checking something, so they're not really actually contributing. So in all those 3 cases, we need to be able to now do some attribution of reward back to the to the actor. The way we do that is we assign a number of fitness levels. These fitness levels are dynamically adjusted because obviously when we start off, we don't know what the maximum fitness changes can be for the domain. We obviously calculate the contributions, and so the weighted and we calculate the probability of an actor sort of contributing to a fitness level, and so that weighted probability is the fitness for the actor. The way we actually implement that is we assign an energy to each actor at birth, and that energy keeps decreasing at different levels depending on how much the actor is actually contributing to the solution. Which means that if an actor energy gets to 0, they're removed from the system. If an actor is consistently and always contributing to the solution of the problem, it will stick around because energy is not going to be deducted from it. So that's kind of how it works. If an actor contributes to the betterment of the solution, this is what this equation here is saying, then we actually deduct less energy from it. But if it also is providing an action that does not get picked up, but at that point the fitness of the domain is actually reduced, that means it was trying to do something that we didn't listen to it, but the fitness kind of got worse, and so in that case the delta energy also goes down. So that's one way of preserving actors that are sort of creative or are pulling a different way when the problem is not being solved. So just to show you a few examples of how this system is working, we did some relatively simple experiments to check to see how it scales. The first experiment is an XOR experiment. At its simplest, a 1 XOR is a simple single XOR gate. So 2 inputs, 1 output, very simple dimensionality. The actors are trying to solve that. And of course, they do in almost all cases within the allotted number of domain actions. So this 100,000 is the number of domain actions allowed for that particular experiment. Now what we do is then to increase this to 2 XORs in parallel. And there's the reason we do this is we're actually increasing the dimensionality of the problem. And the 2 parts of the problem are completely independent from one another. So what we expect is that you cannot solve this with a single actor. It has to be solved collaboratively. So if we go to 2 XORs or 3 XORs, we can see that in most cases, the problem is still being solved within reasonable time. There's a third outcome that could be possible, which is the entire population goes extinct, and in these cases they didn't. One other thing to note here is that in all of the experiments that I'm showing here, we did not change the hyperparameters. There's no setting change between and across these problems, which is important, and it's only possible because of this independence between the dimensionality of the problem and the solution. We don't have to change anything. The very same ecosystem of actors solves these problems, and no need for any tuning, fine tuning. Here's an example of an actor that solves the XOR problem. Again, it's interesting when we look at these rules to note that this actor does not seem to be solving the entire problem itself. It's part of a bigger picture of solving the problem. It also does some interesting things. For example, it actually changes its linked location. So it's actually maybe searching for areas in the environment for some state or some other actor. So this I thought was interesting behavior to show here. And as I said, it shows that the behavior is collective. And how do we know that? You can see here that the impact contribution probability for this actor is 1, which means that it has always contributed to the solution with a probability of 1. However, when you read the rules, it's not solving the XOR problem entirely. So it's part of a bigger picture collaboration to solve the problem. We ran this very same system with no changes to hyperparameters on some simple OpenAI gym problems. Again, mountain car, lunar lander, cart pole, acrobat, and in all these cases, it seems to solve the problem more often than not, and within reasonable time. And so just to show you how the population dynamics work here. This is interesting. This is one of the runs. And the way the run is going forward, this is, I think, an acrobat. No, sorry, it's cart pole. And you can see that there are certain fitness levels that are being discovered by the system, because as I said, the system initially doesn't know what maximum fitness it can achieve. When it does achieve these fitness levels, for example, here or here, the population seems to stabilize and reproduction goes down to 0 almost, which means that it thinks that it solved the problem. But there are perturbations in the system that cause it to kind of snap out of this, maybe an experience of a higher, these are rolling averages, so it might have actually experienced a higher fitness level somewhere here and noticed that it's not quite where it needs to be. And so, you know, we then have some change, but after stabilizing, it actually finds new fitness levels and ultimately solves the problem. Another set of experiments that we did was in actually switching the domains, abruptly switching between domains. We gave the environment a set number of, I think in this case, I'm not sure, it might be 100,000 time elements to solve the problem, and then we switched abruptly to a higher dimension problem and then back. So we actually went from 1 XORs to 2 to 3 and then back to 1. You can barely see that, that's the yellow here. And you can see in most cases it was able to actually solve the problems, all 4. The other thing to observe is that typically it actually solved the 1 XOR problem when it came back to it faster than it did in the first place. And I don't have this shown here, but it actually ends up solving 2 and 3 XOR problems typically faster than it does when it starts from scratch. This dashed line means that it was not able to actually solve, in these cases, the 3 XOR problem within the allotted time. Again, no changes to hyperparameter, an abrupt change to the domain, and you know, the same environment is kind of surviving these huge changes. To compare this to direct evolution, if we had each actor be able to see all the domain states and be able to issue all the actions in the domain and just act as a typical genetic programming kind of direct evolution system, you can see that it's actually in all cases unable to withstand this change and solve the 2 XOR or 3 XOR problems. We ran this in some other permutations of OpenAI gym problems, such as cart pole to mountain car, and in some cases it actually solved the problem. In some cases, even if it was not able to solve the first problem, which is cart pole, it was actually able to then solve the mountain car problem. This is a better example where we went from acrobat to lunar lander, and in all cases, it was able to solve both problems. So to finish off, the main contribution was this common mapping between domain and ecosystem of actors. And so there's this domain independence that this kind of setting provides us. The same system with no hyperparameters, at least in these experiments that we ran, was able to solve a variety of problems and withstand major changes in the problem space. And it is robust to changes in the state and the action space because of this independence. And we are observing some interesting collective behavior that emerge from these actors. Some of the work that we want to do going forward is it would be interesting to design some benchmarks for these types of domain-independent problem-solving settings. We do want to make the system more parallel and asynchronous in design. And we want to add more actor types. The interesting thing here would be to actually experiment with new actor types and see how well they can do in these settings. That's it. I'll take any questions that you guys might have.
Thank you so much, Damak. So we're running a bit late for questions, but what I suggest is we take them to Slack so we can continue discussing. I have a few questions about the scaling myself. Very interesting work, really. Thank you so much for this very nice presentation. And that's the end of our session. Don't forget that we'll have a keynote coming up soon. David Abura today will be presenting about climate change, coral reefs and things. And do we have a message from ALife 2022?
No, it's perfect, correct.
All right.
It's David speaking.